Assalamu alaikum wa rahmatullahi wa barakatuh. Dear students, today inshallah we're going to talk about the epithelial tissue. First of all, we said that the cell is the structural and the functional unit. Cells joined together form tissues, tissues form organs, and organs form system. We have four basic tissues, which are the connective tissue, epithelial tissue, muscle tissue, and nervous tissue. The epithelial tissue, again, these are the four tissues in our body: the epithelial tissue, muscle and nervous, and connective tissue, which include the connective tissue proper, bone, cartilage, and blood. Here, as you can see, that the epithelial tissue is widely distributed in our body. Second point here in this diagram shows that every part of the lining epithelium differ according to the site in order to perform certain function. The general characteristics about the epithelial tissue: they are crowded cells, arises from the three germ cells, which are the ectoderm, endoderm, and the mesoderm. This again shows that the epithelium is widely distributed. The epithelium is avascular, takes its nutrition by diffusion from the surrounding tissues, which is usually the underlying connective tissue. It rests on a basement membrane. It has a high power of regeneration in order to replace the shedded superficial epithelial cells. It either covers surface or line cavity. It is either simple, one layer, stratified, more than one layer. They sometimes form glands which act as a glandular epithelium, sometimes contain receptors, so it's called neuroepithelium, and sometimes has contractile function, and this is known as myoepithelium. Again, the epithelium resting on the basement membrane. They are either surface or lining, or a neuroepithelium or myoepithelium. The surface epithelium either simple or stratified, glandular or neuroepithelial or myoepithelial. So the surface epithelium either simple or stratified, glandular epithelium form gland, and neuroepithelium modify and act as a receptor. The myoepithelium modify so that they have a contractile function. The simple epithelium is formed of one cell layer resting on basement membrane, stratified two or more cell layer. These are some sort of definition. Squamous means that the width is more than the height, thus to say it is flat. Cubical, the width and the height and the same, so they are cube in shape. Columnar, the height is more than the width. This is a diagram showing you different cells shape. This is squamous, this is cubical, and this is columnar. The simple epithelium is divided according to the height and the shape of the cell into simple squamous, simple cubical, simple columnar ciliated, and pseudo-stratified columnar epithelium. First, we're going to talk about the simple squamous epithelium. It is formed of one layer of flat cell with flat nuclei. When you see it in the top view, it appears as if it is fried egg in a pan. The function, it gives smooth surface, and it is delicate for gas exchange. Smooth surface for movement of the blood in the blood vessel and movement of the mesothelium, which is pleura, pericardium, peritoneum, and delicate for gas exchange and glomerular filtration. So it is present. This is the fried egg appearance in a pan. The nucleus is the egg yolk, and the cytoplasm here is the white part of the egg. As we said, for easy movement, it's present in the mesothelium, either the peritoneum, pleura, pericardium, lining the blood vessel for easy passage of the blood. This is the blood vessel lined with simple squamous epithelium. This is S section and this is TS section. Lung alveoli for gas exchange, and glomerular filtration in the Bowman capsule. Simple squamous epithelium, please remember two words which are smooth surface and delicate for gas exchange or for rapid glomerular filtration. Again, this is the mesothelium lined with simple squamous epithelium. The simple cubicle, it is one layer, the length equal to the width and the central nuclei. One layer, cube in shape, the length equal to the width and the nucleus is central and rounded. Please remember two words, secretion and reabsorption. Here is the thyroid follicle lined with simple cubical epithelium. This is the kidney tubules lined with simple cubical epithelium and the function is reabsorption. Here is the glandular epithelium lined with simple cubical epithelium. The part that produces the secretion is called the acini and the part that releases the content is called the duct. Again, these are the simple cubical epithelium. Simple cubical epithelium. The arrowhead points to simple cubical epithelium, while the arrow points to simple squamous epithelium. Again, this is simple squamous epithelium lining the glomerular capillary. This is simple cubical epithelium lining the collecting tubule. Simple cubical epithelium lining the kidney tubules. Lining the kidney tubules. A simple columnar, it is one layer, the length more than the width, and the nucleus is basal and oval. It is elongated cell with elongated, one layer with the length more than the width with basal oval nuclei. Please remember two words, secretion and absorption. So it is present in the small intestine and in the stomach. Secretion as in the stomach, small and large intestine for absorption. It contains microvilli to increase the surface area. This is simple columnar epithelium. As you can see here, tall cell with basal oval nuclei. Again, this is simple columnar epithelium, one layer of tall cell with basal oval nuclei. Remember two words which are the secretion and the absorption. Secretion and absorption. Present where again in the gallbladder, present in the main pancreatic duct and the common bile duct. What the meaning of goblet cell? It is modification of the columnar cell in which it accumulates mucus at the surface. This mucus is important, act as a lubricant for the stool in the small and large intestine, and is important in the respiratory passage to trap the particles. The apical part expands and is full with mucus, while the basal cylindrical and contain the nucleus and the organelles. Again, this is simple columnar and these are the goblet cells. Again, these are the columnar. These are the columnar cell and this is the goblet cells. As you can see, the apical part is distended with mucus and the basal part here contains the nucleus. Simple columnar ciliated epithelium, one layer. The cells are tall with basal oval nuclei and the surface contains cilia. What I want from the cilia is movement in one direction. So it is present lining the fallopian tube in the uterus and the lower respiratory passages as the lung bronchioles. The free surface contains cilia present in the uterus, fallopian tube, and the lung bronchioles. What is the function? The cilia transport fluid or the ova over the epithelial surface in one direction. Again, this is simple columnar ciliated epithelium. Here is the simple columnar. This is simple columnar ciliated. This is scanning electron microscope for ciliated epithelial cells. Ciliated epithelial cells. What do I mean by pseudostratified columnar epithelium? The word pseudo means false False stratification, so it is simple. False stratification, so it is simple. It is one layer of crowded columnar cell. White appears as if it is stratified due to the crowdedness of the epithelial cells and the nucleus present at different levels. But the fact it is simple. Why? Because all of them rest on the same basement membrane, but not all of them reach to the surface. So I can see nucleus at different levels, so I said it is stratified. But it is false stratification. It is a simple. As you can see here, I am having two levels of the nucleus, but in fact all cells rest in the same basement membrane, but not all of them reach the surface. That do not reach the surface. Pseudostratified columnar non-ciliated present where the site present in the male genital tract as membranous male urethra and in the vas deferens. While pseudo-stratified columnar ciliated with goblet cell present where present in the upper respiratory passage. What is the function? Sweep out mucus trapped particles. Sweep out mucus trapped particles. Present in the upper respiratory tract, nasal cavity, trachea, and bronchi. Here are the cilia and these are the goblet cell. Pseudo-stratified columnar ciliated with immotile cilia. In fact, it is long microvilli. It present in one organ in the male genital system in the epididymis. This long microvilli is important. Why? Here the sperms are stored. During this period, there is change in the morphology of the sperm and absorption is needed. Pseudo-stratified epithelium. Pseudostratified, and this is pseudostratified columnar ciliated with goblet cell. This is pseudostratified columnar, for example, present in membranous urethra or in the vas deferens, and this is pseudostratified columnar ciliated with goblet cell, present in the trachea, in the bronchus, in the nasal cavity. Again, pseudostratified columnar epithelium. This is present in the ducts system of the salivary gland. Pseudo-stratified columnar ciliated with goblet cell. Stratified epithelium, the main function is protection. What do I mean by stratified epithelium? More than one layer. The basal cell lying on the basement membrane, it is mostly columnar or cubical in shape. The intermediate are polyhedral and the most superficial differ according to the type. We said if the most superficial is squamous, so it is stratified squamous. The most superficial is cubical, so it is stratified cubical. The most superficial is columnar, it is stratified columnar, and another type known as the transitional epithelium or the cuboidal epithelium. In the surface are cuboidal cells. This is the stratified squamous epithelium. First, it lies on a wavy basement membrane. The first layer is columnar with basal oval nuclei. The other layers, the middle layer, formed of polyhedral cell with central rounded nuclei, and the most superficial cells are flat cell with flat nuclei, which are squamous. It is the thickest type of the epithelium. It may reach to 30 layers. Lie on a clear wavy basement membrane. Why there is wavy basement membrane? For two reasons. To increase the surface area in order to supply the epithelium with more nutrition. As we said before, any folds means or any waves means that I am increasing the surface area. Increasing surface area for what? For nutrition. Number two is to fix the epithelium to the underlying tissue. The first layer are columnar with basal oval nuclei, and it is active and continuously dividing in order to replace the shedded most superficial cells. The middle cells are polyhedral. The word polyhedral means many sides with central rounded nuclei. The more we go upward, the more the cells decrease in size. Why? Because as we go upwards, I am away from the nutrition, which is underlying connective tissue. The superficial cells are flat cells with flat nuclei. We have either non-keratinized, not covered with keratin, as What? Present in the wet surface, mucous membrane as the oral cavity, esophagus, vagina, tips of the urethra, anal canal, cornea, and conjunctiva. Stratified squamous epithelium, non-keratinized, not covered with keratin. Keratinized stratified squamous epithelium. Here the epithelial tissue is covered with a layer of keratin. It's covered with a layer of keratin. It is the strongest type of epithelium. Their superficial layers are away from nutrition, so they die, form keratin. Those the cytoplasm and the nucleus only contain keratin, which is important for protection. Present in the epidermis of the skin and present in the dry areas of the skin as the skin and what open upon it as the external ear, external. The transitional epithelium, by its name, I am decreasing in the thickness of the layers from a filling or empty state in the bladder. In empty, it appears to be 6 to 8 layers resting on clear basement membrane. The basal cells are cubical with rounded nuclei, and the intermediate polyhedral with round. Here is, dear student, transitional epithelium photomicrograph of the transitional epithelium. Here the basement membrane, it is not clear. From the basal part to the apical, from the basal cell to the apical, all the cells are rounded nuclei. Take care that the most superficial cells are cuboidal in shape and some of them binucleated. The transitional epithelium, dear students, the most superficial cells are a large dome-shaped cell. Sometimes are binucleated, covered by something called dense plaque that protect it against the action of urine, and the most superficial cells are connected together by junctional complex. In the expanded state, the cells glide, have the epithelium compressed and stretched without damage and become thinner to allow distension in the bladder. It is present in the renal pelvis and ureter-urinary bladder. Here is the empty bladder and here is the full bladder. In the full bladder, stretch occur, so the cells, as you can see here, the cell as if they are gliding over each other. Okay. So in the stretch stage of the in the full bladder, as if the number of cells decrease, but actually the thickness of the cells decrease. Transitional epithelium or uroepithelium, as you can see here, non-clear basement membrane. All the cells are rounded with all of them consist rounded central nuclei. And the most superficial cells are large dome-shaped cell or cuboidal cell. As we said, it's present in the ureter, in the bladder, and the proximal part of the urethra. The function is distensibility. Then this is stratified cubical columnar epithelium. The most superficial cells are columnar cell, and it is either non-ciliated or ciliated. It is present, the stratified columnar, similar to the stratified squamous, basal cell columnar, then polyhedral with central rounded nuclei, and the most superficial cells are columnar. They are either non-ciliated, as present in the benign male urethra and the rectoanal junction, and ciliated only present in the fetal esophagus. Stratified cubicle is formed of 2 layers of cubical cell, and this is found only in the duct of the sweat gland. Stratified cubical epithelium, 2 layers of cubical cell. This is stratified columnar cell. As you can see here, the most superficial cells are columnar. This is the stratified squamous epithelium, non-keratinized, and this is the keratinized stratified squamous epithelium. This is the transitional epithelium. This is again non-keratinized stratified squamous epithelium, and this is keratinized stratified. This is non-keratinized stratified squamous epithelium, and this is the keratinized stratified squamous epithelium. This is the non-keratinized stratified squamous epithelium, and this is the keratinized stratified squamous epithelium. This is again non-keratinized stratified squamous epithelium, and this is the non-keratinized stratified squamous epithelium. Again, this is keratinized stratified squamous epithelium, and this is transitional epithelium. This is keratinized stratified squamous epithelium, and this is non-keratinized stratified squamous epithelium. Now we're going to talk about the glandular epithelium. It is divided into two main subgroups, either exocrine gland, acini and duct, or endocrine gland cells, and release their content through the blood. Due to presence or absence of the duct, we have an exocrine, in which we have an acini and excretory duct, while the endocrine we have cells and blood capillaries. There is no duct system. Mixed as, for example, the pancreas, exocrine part which secrete the pancreatic enzyme and endocrine part which secrete the hormones as islets of Langerhans. The classification of the exocrine gland has many classifications. First of all, as we said, number of cells, for example, unicellular as goblet cell, multicellular as salivary gland. According to the nature of secretion, either watery, serous, mucus, mucoserous. According to the mode of secretion, no change occurs as merocrine. The apex released with the secretion as the mammary gland. All the cell rupture holocrine. This is in sebaceous gland. As you can see here, dear students, this is diagram showing the different mode of secretion. Here, all the cell rupture, so this is the holocrine. Here, no change occurs. The cell releases the content by exocytosis, so it is merocrine. Number C, the apex is dislodged with the secretion. This is apocrine. So this is number A, holocrine, B, merocrine, C, apocrine. According to the function, either secretory, excretory, according to the branching of the duct system, simple, not branching, simple branched, just two branches, compound, many branches. According to the shape of the secretory unit, either tubular, acinar or alveolar, or tubulo-alveolar. This is diagrammatic picture for simple tubular. Simple means I'm not branching. Tubular, the shape is tube. This is branched, simple, branched, tubular. This is simple, coiled, tubular. This is simple, alveolar. This is branched, alveolar. And this is compound tubular, compound alveolar, and compound tubulo-alveolar. What do I mean by compound tubulo-alveolar? The secretory portion, tubular and alveolar in shape. According to the branching of the secretory, we have said and branched. This is simple tubular as present in the small intestine, called tubular as present in the Schwann gland, alveolar as present in the sebaceous gland. Branched, tubulo-alveolar is not present, simple tubulo-alveolar not present in human. Branched tubular as small intestine, simple branched tubular as in the stomach, fundus of the stomach, branched alveolar as in sebaceous gland. Branched tubulo-alveolar as in the labial glands. Compound exocrine glands. We have either compound tubular as in the liver, kidney, and testis, compound alveolar as in the mammary gland, compound tubulo-alveolar as in the pancreas and the main salivary glands. Okay, these are many examples of the glandular epithelium. Here is the pylorus, which is branched tubular. Okay, here is the testis, compound tubular. Here is the sebaceous glands. This is the labial glands. This is the mucoserous glands. Again, this is mucoserous glands. Okay, serous glands. mixed gland, exocrine and endocrine as the pancreas. All these are examples, and you know, in the exam, the part of the examples of the glandular epithelium is cancelled. The example of the glandular epithelium only. Glandular epithelium only. I'm stress on example of the glandular epithelium. What about the neuroepithelium? The epithelium is specialized to act as a receptor. Example, the taste bud that present in the tongue, soft palate, pharynx. The epithelial tissue modified to receive external stimuli. And the neuroepithelium consists of supporting cells and sensory cells. Found in the tongue as taste bud for taste, present in the nose for smell, present in the internal ear for hearing, and present in the retina for vision. Here is a taste bud this is tongue papillae, known as circumvallate. You're going to take it inshallah in the second year. These are the taste buds that will receive taste sensation, that will send it to the brain. Again, this is a taste bud which is formed of supporting cells, sensory cells, and stem cells. Here is the epithelium. Here is the epithelium in the olfactory epithelium, pseudostratified columnar, ciliated, with no goblet cells. Inner ear receptors, we have receptors for hearing, known as organ of Corti. Vestibular receptors needed for the equilibrium as macula utricle, macula saccule, and crista ampullaris. These are names for receptors in the inner ear. This is the organ of Corti that is responsible for hearing. These are the crista ampullaris and utricle and saccule that present needed for equilibrium. This is the retina. Myoepithelium is a specialized type of epithelium modified to contract. As you can see here, this is the acini, and this is the myoepithelial cells. The myoepithelial cell branched, and when they contract, they squeeze the secretion that move directly to the duct. So they are specialized type of epithelium. The cell branch into multiple processes surrounding the secretory portion. Help to squeeze. When they contract, they discharge the content into the lumen, then to the duct. Present in the sweat gland, salivary, and mammary glands. Now we are going to take what's so-called epithelial polarity. What do I mean by epithelial polarity? These are cells that present neighboring to each other or in contact with each other. Here is the lumen. So there is something called apical modification, and this is the base known as basal, and between adjacent cells something called the lateral modification. The apical faces the lumen or outside the world, separated from the basolateral side by tight junction. The basolateral that face neighboring cells, the basal that adhere to the extracellular connective tissue basement membrane. Apical region, we might have microvilli to increase surface area, stereocilia, which is elongated microvilli needed for absorption of the content, and this is present only in the epididymis. Cilia that move in one direction. The microvilli is projection contain actin filament in the center. The microvilli increase the surface area for absorption. Here is light microscopic picture of the microvilli. This is what's called terminal web. Here is the microvilli formed of actin filament covered with the cell membrane. Stereocilia, it is not true cilia. As you know, the cilia is long, and the microvilli is short. Here I see something long, so I called it cilia. But actually it does not move, and it is formed of actin filament, so it's called stereocilia or long microvilli. These cilia are motile process present on the surface, move in one direction. It's formed of the basal part, which is equal to the centriole, formed the basal part of the cilia is formed of 9 triplets. The shaft is formed of 9 doublets and 2 singlets in the center. The lateral specialization, which is the intercellular junction. Here is a cell and here is a cell. What's present between the cell is called lateral specialization. First, we're going to take tight junction. Tight junction or called occludent junction. Tight junction or called occludent junction. We have something called zonula occludent. We have something called the zonula occludent. Meaning what? Zonula means pellet-like. Adjacent cell membrane fused together. There is no space between the cell. The space is 0 between the cell. So this is one cell and this is another cell. The space between them here is 0. And the tight junction is present as a pelt. Okay? Completely encircle the apex as a pelt, form a seal or prevent the passage of fluid in between the cells. As you can see here, this is one cell and this is another cell. There is no space present between two cells. The zonula adherence, as we said, zonula means a pelt-like, but there is a space between two adjacent cells, reach up to 20 nanometer. There is intercellular space filled with adhesive materials. This adhesive material is the cell coat that present of two adjacent cells. It is encircled the whole cell as a pallet. The cytoplasmic site of the membrane contains dense protein in which actin filaments are inserted. It fixes cells together. So the adherence junction or zonula adherent, the meaning of zonula, belt-like. There is a space about 20 nanometer between two adjacent cells. In this space, there is adhesive material and the cell coat of the two adjacent cells. On the cytoplasmic aspect, there is secant protein that actin filaments are inserted on it. The function is to fix adjacent cells together. This mosome or the macula adherens, the macula means point. It is a limited or spot not encircle the whole cell with a space 30 nanometer. In the membrane, between the two membranes, there is transmembrane linkers, proteins, and the cytoplasmic aspect thickened. Attached to the cytoplasmic aspect, turnofilaments, which are intermediate filaments, okay, inserted as hairpin. It fixes cells together. Again, the macula adherens, the meaning of macula means spot. Only a spot, not a pelt-like, not zonula. Okay, there is intercellular space. It's about 30 nanometer, and there are linker proteins. On the cytoplasmic aspect, there is dense protein inserted to each intermediate filament in the form of a hairpin-like attachment. It keeps cells attached together, fixes cells together. The fourth type of junction is known as gap junction or nexus. There is a narrow gap crossed by communication channels. Each is formed of 6 proteins. Allow free communication between two cells. Passage of ions or impulse or small molecules. Found between smooth muscle, cardiac muscle to allow rapid passage of impulse. This is a cell and this is another cell. And these are the proteins that form the connexin, the channel. This is the connexin, okay? 6 proteins 1, 2, 3, okay, and from the other side 1, 2, 3, forming a channel. Here are the tight junction. Here is the zonular entrance. Here is the desmosome, and here is the connection. What is this? It is one of the basal modifications that we're going to talk about later. Again, this is a cell, and this is another cell. This is tight junction, and this is the, here is the desmosome. Here is the desmosome. Okay, linker proteins, dense plaques, and intermediate filament attached on it as a hairpin-like. And these are gap junction. Lateral specialization, I'm going to talk rapidly about it again. We have something called junctional complex. I have more than one junction. First, I'm having tight junction to prevent passage of fluid between adjacent cells. The meaning of junctional complex, I'm having more than one junction present between two adjacent cells. The best example in the columnar cell lining the small intestine. First, we have zonula occludent at their top, followed by zonula adherens, and then desmosome and gap junction. What the meaning of a junctional complex? I have many types of junction. First, I'm having tight junction, no space between the cell. Then I'm having zonula adherens, then desmosome, then the gap junction. Now we're going to talk about the apical specialization. We have the microvilli, which is finger-like projection. They contain actin filament. They form what's called brush border. By EM, having actin filament covered by cell membrane, its importance to increase the surface area is present in renal tubules and small intestine. This cilia is long, hair-like projection. They are motile, formed of microtubule. It's formed of the shaft of the cilia is formed of 9 doublets and 2 singlets, present in the upper respiratory tract and in the fallopian tube. Stereocilia, as we said, it is not a cilia, it is long microvilli, and it is not motile, present in only one space, which is called one place called the epididymis. It's important for absorption of the content of these, as the epididymis store sperm, and many changes occurs in this sperm during which absorption is needed. Flagella, it is the long cilia, which is the tail of the sperm. It moves like whip-like movement in order the sperm move in the female genital system. And now we are going to talk about the basal specialization. Now we are going to talk about the basal specialization. Here is the basement membrane. The basement membrane is boundary between the epithelium and the underlying connective tissue. It's stained with silver, appears brown, bypass appear red. It is clear and thick in the epidermis, non-clear in the urinary bladder. By EM, it is formed of the basal lamina and the reticular lamina. The basal lamina, the part related to the epithelium, while the reticular lamina, the part related to the underlying connective tissue. What is the function of the basement membrane? Bind the epithelium to the underlying connective tissue, help the epithelial growth and repair, in the glomerular capillaries act as selective barrier. The molecular structure, the basal lamina, which is related to the epithelium, is formed of collagen type 4 and collagen type 4 and the cell coat of the epithelium, while the reticular lamina is formed of collagen type 3, which is the reticular fiber. There are collagen type 7 anchoring fiber anchoring the basal lamina with the underlying connective tissue. The basal specialization here again, in addition to the basement membrane, there's something called the hemidesmosome. What the meaning of hemi means half the desmosome. It is formed between the basal part of the cell with the basement membrane. It fixes the basal part of the epithelium to the underlying connective tissue. It is similar to the desmosome, but it's half desmosome. Only I'm having dense plaque and intermediate filament here as a hairpin appearance. Basal infolding, found in the epithelium, specialization in ion transporting cells as though present in the renal tubules. What I can see here is basal infolding of a cell membrane. In between them there is longitudinally arranged mitochondria. The basal membrane formed of deep invagination, dividing the basal compartment the basal part into compartment, contain vertically oriented mitochondria, and the function of which is ion transport. So it's present in renal tubules. Again, with the meaning of junctional complex, more than one junction. I can see at the apex, the best example, cells lining the small intestine. Here at the apex, I'm having zonula occludens, then zonula adherens, then macula, which is the desmosome gap junction. Again, this is diagram for the desmosome. Here, the space about 30 nanometer between two cells. Here are from the cytoplasmic aspect dense plaques, and intermediate filament act as a hairpin-like. Okay, this is an EM picture of the desmosome. You can see the intercellular space contain the linker proteins. Here are the intermediate filament connected to the dense proteins on the cytoplasmic aspect. Here is an electron microscope of the junctional complex between two cells. First we have the tight junction or zonula occludens, then we have zonula adherens, and then the desmosome. This is junctional complex. Again, in the basal region we have the basal lamina. In the basal region we have basement membrane, hemidesmosome, and basal infolding. The basement membrane is formed of the basal lamina and reticular lamina. As we said, the basal lamina is formed of collagen type 4 and protein, protoglycan and glycoprotein, and this is secreted from the epithelial cell, while the reticular lamina is formed of collagen type 3, which is reticular fiber, and there is anchoring fiber connected the underlying connective tissue with the basement membrane. And we said, what about the hemidesmosome before? Again, this is cilia, okay, and this is microvilli. This is basal infolding, okay, and these are the mitochondria. Again, these are basal infolding, and these are mitochondria. This is junctional complexes, zonula occludens, zonula adherens, and desmosome. This is the hemidesmosome, connecting the basal part of a cell with the underlying connective tissue, formed of the dense plaque here and intermediate filaments. What is the function of the epithelium? We have protection as present in the epidermis of the skin, secretion as in glands, absorption as cells lining the small intestine, excretion as cells in the kidney, sensation as neuroepithelium, reproduction as that in the gonads, ovary, and testes, producing the ova and the sperms, smooth lining and that lining the blood vessels. Thank you, dear.